The next part of this unit is about cells, and so we need to do a little bit of review about cell organelles and perhaps cover some material that you haven't had before in talking about cells. One thing I do want to point out to you that the first part of Chapter 6 is about microscopy, and although I'm not doing any lecturing on microscopy, you need to be familiar with the different types of microscopy and what they can be used for. There are several different techniques that are discussed in this part of the chapter. Several different light microscopy techniques as well as electron microscopy techniques, and so be familiar with those things because that could be relevant to a lot of things we do in the future. But let's begin now talking about cells. Remember that the cell is the fundamental unit of life, and all living things are made of cells, the main part of the cell theory from way back when. The cell is the first unit where life can be identified. In other words, life is emergent property when you get to the cell level. And the structure of cells can vary there are so many different kinds of cells, I can't even start to tell you how many there are, but the structure of each cell is correlated by their function. Some of them need to be long and skinny, like neurons. Some of them need to be short and fat, like glial cells. They have all different kinds of functions and all different kinds of structures, and so we'll talk some more as time goes on about those different kinds of things. Cells, of course, are all related to each other by their descent from earlier cells. Remember, all cells come from previously existing cells, another important part of cell theory, and we'll continue on talking about that. Eukaryotic cells have internal membranes that compartmentalize the functions in the cell. Remember that we have two basic kinds of cells. We have prokaryotic cells and eukaryotic cells. The prokaryotes, of course, are only organisms that belong to domain bacteria and domain archaea, and all other organisms belong to the eukarya, which have eukaryotic cells. Now, basic features that all cells have in common, of course, plasma membrane, the cytosol, also called cytoplasm, when it's got the other stuff in it, chromosomes, and ribosomes. Prokaryotic cells are different from eukaryotic cells because they don't have a nucleus. Their DNA is not bound up by a membrane. It's just in the central region of the cell called the nucleoid. There are no membrane-bound organelles, and the cytoplasm is bound by the plasma membrane, but there are no internal membranes in the cell. Here is a basic bacterium. This is Bacillus coagulans. Transmission electron micrograph right over here shows you the same basic structures. We have some bacteria have fimbriae, which are kind of like Velcro on the surface. A lot of them stick to things. Some have flagella. Not all of them do. This particular diagram on the left has one. The nucleoid is the region where the DNA is located, and of course that's the bacterial chromosome, generally considered to be a loop of DNA. Although you can see here it's not just a simple loop, it's a big loop. There are ribosomes in bacteria. And bacteria also have a cell wall. Many of them also have a capsule, which is it's a slimy or sticky coating on the outside. And so they're just basic cells. They don't really have a whole lot of structure to them. There are different kinds of prokaryotes, of course, different shapes, different species, different sizes, but we'll talk a lot more about bacteria later on when we talk about microbes. We're going to focus the rest of this part on the eukaryotic cell. So when we compare prokaryotic and eukaryotic cells, you can see that the eukaryotic cells are different because they do have a nucleus that is bound by the nuclear envelope. They have other membrane-bound organelles, and the cytoplasm in the region between the plasma membrane and the nucleus is referred to as cytoplasm. Actually, it's also called cytoplasm in the bacterial cell as well. The cytosol is the fluid. Cytoplasm is inside the cell containing the parts of the cell. Generally speaking, eukaryotic cells are generally much larger than prokaryotic cells. There are some very small eukaryotic cells and some very large ones, as well as very small and very large bacterial cells. But generally speaking, the eukaryotic cells are a great deal larger, on the order of 100 to 1,000 times larger than prokaryotic cells. The plasma membrane, which is also present in the prokaryote, okay, is this barrier that allows certain things to pass through and others not. We call it selectively permeable. Generally speaking, it is a double layer of phospholipids with embedded proteins, various kinds of proteins. Remember, we've got the hydrophilic region of the phospholipid molecules, that is the head region, and then the hydrophobic tail regions, and the bilayer is oriented so that the hydrophobic region is in between the two hydrophilic regions. We have various kinds of membrane proteins here, integral proteins as well as transport proteins. A lot of these are glycoproteins having carbohydrate side chains and so forth. You can see in a transmission electron micrograph, you can see the double layer of the plasma membrane. Now, cells have different sizes, of course, and they do grow throughout their life. Remember from your first biology course that there are metabolic requirements that limit the size of cells. Remember, there comes a time when it's the surface area to volume ratio decreases to the point where it is difficult to transport needed materials in and out and to get rid of waste materials. And so it's related to the surface area to volume ratio. You can see here the mathematical construct of that. As the surface area increases, it increases by a factor of n squared, but the volume increases by a factor of n cubed. So it's much more efficient to remain relatively small rather than large because you have a greater surface area to volume ratio, and that allows for the appropriate passage of materials through the membrane to nourish the cell and take care of the other metabolic needs of the cell. We will do a lab soon that talks about, that goes through some of these things about the cell size and calculating the rate of diffusion to see how that is affected by size. Here we have a generalized cell. This is a generalized eukaryotic cell, probably an animal cell. And so it shows lots of different structures here. We'll talk about all these in turn. You can see all of the different structures, the nucleus, the endomembrane system, including the rough and the smooth ER, the Golgi apparatus, mitochondria, peroxisomes, ribosomes, both free and bound. We can see some cells have a flagellum, like this one shows a flagellum there. Some have also microvilli, which have increased their surface area. There are also lysosomes, which are kind of the digestive system of the cell. And then we've got the cytoskeleton, which is composed of 3 different kinds of filaments, the microfilaments, intermediate filaments, and microtubules. And we'll talk about each of these in turn. Plant cells have many of the same structures. They do not have lysosomes, but they do have chloroplasts with an internal membrane system. But all the other structures that we talked about in the animal cell are also present in the plant cell. So let's start with the nucleus and the endomembrane system. The nucleus, of course, is the place where most of the genes are located, and it is the most conspicuous organelle inside the cell. When we look inside a cell with a light microscope, usually the nucleus is the most recognizable thing that we can see. The other organelles are enough smaller that it's difficult to see them with the light microscope. The nuclear envelope is the double membrane structure that encloses the nucleus and separates it from the cytoplasm. Each one of those membranes consists of a lipid bilayer, so really there's two bilayers surrounding the nucleus. Here we can see various structures that you see in the nucleus. We've got the nucleolus, which is a denser structure inside there. The chromatin, of course, is the DNA and proteins that make up the chromosomes when they condense. There are pores in the nuclear membrane that control what goes into and comes out of the nucleus. Not everything can go in and come out. The rough ER is very closely associated with the nuclear membrane. In fact, it's pretty much contiguous with the nuclear membrane here. We've got inside the nucleus, we've got a nuclear lamina, basically, which is a series of protein fibers that kind of helps organize it. There's probably also a nuclear matrix, which also helps keep the chromatin in a position that it can easily be translated or transcribed into messenger RNA. Here we see the chromatin. That's just the DNA surrounded, wound around these histone proteins. And again, we'll talk more about that later on. The pores regulate what comes into and goes out of the nucleus. And then the shape of the nucleus is maintained by that lamina and probably also a nuclear matrix. There's evidence to show that there's probably a matrix in there. I don't think that's definitive at this point in time, but both of those things together may help organize the DNA and maintain the shape of the nucleus. Inside the nucleus, there are units of DNA called chromosomes, of course, and that's composed of DNA molecules associated with proteins, wound around those histone proteins. And together, those things are called chromatin. Remember that also you don't see the chromosomes as separate bodies inside the nucleus except when it's time for cell division. Once it's time for cell division, then the DNA or the chromatin condenses into the chromosomal bodies that are then visible after the nuclear membrane dissolves when the cell is dividing. The nucleolus is also located inside the nucleus. This is a denser body inside the nucleus. It's not always visible in all cells, although I think most cells have a nucleolus. This is the site where the ribosomal RNA is synthesized. The ribosomes are made of ribosomal RNA and associated proteins, and these are the sites where proteins in general are constructed. There are both free ribosomes in the cytosol as well as ribosomes that are bound to the surface of the rough ER. There is evidence to show that they can move back and forth between free in the cytoplasm and bound to the ER. The ribosomes are not considered organelles because they're not membrane bound. They're just these bodies that have a structure that allows for translation to take place. They're composed of two subunits, a large subunit and a small subunit, and the messenger RNA basically attaches through here, and inside, in between there, is where the reactions occur, the enzyme catalyzed reactions to the dehydration reaction that add those amino acids together to make the peptide bonds. So you'll see ribosomes throughout the cytoplasm of cells. The endomembrane system is really in charge of regulating the traffic of proteins, and it also performs other needed functions, metabolic functions in the cell. So the things that you find as part of the endomembrane system are the nuclear envelope, the endoplasmic reticulum, both rough and smooth, the Golgi apparatus, lysosomes, vacuoles, and the plasma membrane. And these are either continuous or connected by a transfer by vesicles, because some of them are connected to each other. The nuclear envelope and the rough ER and the smooth ER are connected, but then they also can transport to other structures like the Golgi apparatus with vesicles that can pinch off of the ER and travel through the cytosol to the other organelles. So let's look at the endoplasmic reticulum first of all. This is the factory basically where the proteins are made, and this accounts for more than half of the total membrane in most eukaryotic cells. It is continuous with the nuclear envelope, and there are two distinct regions of ER, the smooth ER, which doesn't have ribosomes, and the rough ER, which does. So here we see this endomembrane system, the nuclear envelope, which is again continuous or contiguous with the rough ER, usually right surrounding the nucleus, and then the smooth ER is usually found a little bit farther away and is more tubular rather than lamellar like this. You can see here there's different structures are named here. The lumen is the opening in between the membranes there. Cisternae are basically the different groups of membranes there for different reactions to happen. And then when part of them pinch off to transport to the Golgi apparatus or someplace else, that's called a transport vesicle. The transitional ER would be toward the outside, and that would be where these transport vesicles would break off. Smooth ER is mostly involved with lipid synthesis, like oils, phospholipids, and steroids, including steroid hormones. It's also involved in the metabolism of carbohydrates. And cells that do detoxification, like the liver and things like that, have lots of smooth ER. So there's some kind of functions that it does in relation to breaking down drugs and poisons. Your book says that part of that reaction involves adding hydroxyl groups onto the molecules and making them soluble. And then there are other enzymes in there that will help break them down. The smooth ER is also a location where calcium ions are stored. Muscle cells have a great deal of smooth ER because they depend on the transport of those calcium ions to change the polarity of the cell membrane, and that's what propagates the nerve transmission from one end of the cell to the other. The rough ER has the bound ribosomes. That's why it's called rough. It looks kind of like sandpaper, okay? And that's where the glycoproteins, the proteins that have carbohydrates bound to them, are produced. It distributes the vesicles that transport proteins in a membrane to another location in the cell or to the Golgi apparatus or for export, direct export from the cell. And it's also where the membranes are actually put together in the cell. The Golgi apparatus is the shipping and receiving center. I often refer to it as kind of like the UPS store of the cell. This is a series of flattened sacs that are called cisterna, and it functions to modify products of the ER. And there are certain macromolecules that are put together there, and then it also sorts materials into transport vesicles for export from the cell. Kind of like when you have a package that you need to wrap and ship, you might take it to the UPS store and they've got the packaging as well as can take care of the shipping for you. Here we have the structure of the Golgi apparatus. There are two surfaces, the cis face, which is the receiving side, and the trans face, which is the shipping side of the Golgi apparatus. They used to think that vesicles transported the materials from one cisterna to the next, and it may still happen in some cases. However, current thinking is that as these the molecules are being changed, basically the cisterna mature and pass through the Golgi apparatus from the cis face to the trans face, and then new cisterna are added on the cis face as needed. This is a transmission electron micrograph of Golgi apparatus. You'll see a lot of Golgi apparatus in cells that are secretory cells, cells that make hormones and other kinds of products that are secreted from the cell. Lysosomes are basically a membrane sac that has enzymes inside of it, and the enzymes are hydrolytic. That means they can carry out hydrolysis, and that will help them break down macromolecules. There are various enzymes that can be found in lysosomes, but they can work to hydrolyze proteins, fats, polysaccharides, nucleic acids. They do their job best in the acidic environment inside the lysosome rather than the more neutral environment in the cytoplasm. So oftentimes a food vacuole that might have food in it would fuse with a lysosome, and that way the enzymes can stay in their more acidic environment. Some cell types can engulf other cells by phagocytosis, and that's where you see this kind of thing happen. Then you can also use the lysosomes to recycle the cell's organelles and macromolecules. That's called autophagy. The phagy part refers to eating, okay? And so basically that's worn out cell parts or things that don't work properly anymore, or maybe at various times in the cell's life it might need more of certain kinds of organelles than other times, and so when they're not needed anymore, they can be digested or broken down or recycled by the lysosomes. Here we see lysosomes, and this takes you through phagocytosis and autophagy here. You can see the lysosomes in this transmission electron micrograph here. A peroxisome is another type of membrane-bound organelle that can break down various molecules by converting them, producing hydrogen peroxide, which can then be easily converted to water. And the peroxisomes have a crystalline enzyme structure inside of them. There are various kinds of vacuoles found in all cells. These are in all eukaryotic cells. And these can be used for lots of different things. They can be used for storage, or they can be used to get rid of excess waste or excess water, like in the case of the contractile vacuoles in protistans. Plant cells usually have a large central vacuole, and that is used oftentimes to help maintain the structure of the cell. It adds some pressure inside the cell to maintain the structure of the plant cell. Plant cells really like to be hypotonic. That means they like to have excess water inside them. And so that would be a useful part of that. Central vacuoles can also be used to hold things like starches and as well as water inside various kinds of plant cells. Animal cells also have vacuoles. They're usually smaller than what you find in plants, but they are present in also fungal cells.